Welcome back, everyone. Today I wanted to look at and analyze the data and evidence behind my hypothesis. I will make it known that I'm not a certified in biology, sociology, etc., but I am a certified peer support specialist in mental health. I also graduated from UTI, where we learn a little bit about physics. I suck at chemistry and I'm dyscalculic, so take that however you will. And let's just jump right into it. Just in case you don't know, my hypothesis is actually three: the latent eusociality hypotheses, the epigenetic suppression hypotheses, and the eusocial restoration hypotheses. All of these are based off of my framework, so unfortunately, I do not have any sources or references on this, except the research from others that follows. The hypotheses. If humans are fundamentally a social species, naturally predisposed to selfless cooperation and collective care, then the observed patterns of individualistic and tribalistic behavior are not our true nature, but are instead phenotypic expressions of accumulated genetic trauma that has prioritized survival-based hypervigilance over prosociality since the divergence of our lineage. Or the expression of human use social traits, such as division of labor and cooperative brood care, is epigenetically suppressed by transgenerational trauma markers. These markers, accumulated through millions of years of environmental stressors, maintain the human brain in a state of individualistic survival mode, preventing the formation of a true superorganism. And finally, humans are biologically eusocial species whose natural capacity for collective operation was distributed by accumulated genetic and cultural trauma. This trauma, dating back to ancient environmental upheavals, triggered the breakdown of our bicameral cooperative state and forced the development of individualistic consciousness as a survival mechanism. Therefore, prioritizing global mental health is necessary biological and cultural intervention to resolve this ancient trauma, allowing the human species to transition from individualistic survival back to a self-aware, intentional eusocial organism. Now, I will present the supporting research. Research into the Gaia hypothesis and collective consciousness intersects at the idea that Earth functions as a single, integrated living entity. While the Gaia hypothesis primarily specific framework for planetary self-regulation, collective consciousness provides psychological and philosophical lens through which we view human interconnectedness with that system. The Gaia hypothesis, Earth as a superorganism, proposed by James Locke and Lynn Margulis in the 1970s, the Gaia hypothesis suggests that living organisms and their inorganic surroundings form a self-regulating complex system to maintain the conditions for life. This system regulates variables like global temperature, ocean salinity, and atmospheric oxygen through homeostatic feedback loops. In the simplest terms, homeostasis is the process by which a system maintains a stable internal state while the environment around it changes. It is the biological thermostat. To answer critics who argued the planetary regulation would require conscious foresight, Lovelock created Daisy World. This mathematical model shows how simple competition between black and white daisies can stabilize a planet's temperature without any conscious planning. So there's two different scientific standings. There's weak Gaia, the widely accepted view that life significantly influences the environment, and then strong Gaia, the more controversial view that the Earth system aims for optimal conditions, often criticized for being teleological, implying a goal or purpose. On the Gaia hypothesis, homeostasis is the central fact that makes this theory work. Here's how it functions across different scales. The biological mechanism in a human or animal homeostasis is a constant balancing act managed by negative feedback loops. If you get too hot, your body triggers sweating to cool down. If you get too cold, you shiver to generate heat. Your body maintains a specific pH in your blood and a specific concentration of glucose. If these drift too far, you die. James Lovelock argued that Earth, the planetary mechanism, exhibits homeostasis instead of brain or nervous system. It uses biogeochemical loops. To compare these three systems, neurology, Gaia, planetary regulation, and collective eusocial consciousness, we could look at the mathematical scales of their components, processing speeds, and connectivity metrics. Relativity dictates that no information, whether a neural impulse or a planetary chemical signal, can travel faster than the speed of light. If neurons fired faster than the speed of light, your brain could theoretically be the size of a planet and still function with the same coordination you have now. Because neurons are relatively slow, biological consciousness is physically limited to small, dense structures like brains, or extremely slow systems like Gaia. If you view Gaia as a global brain, it has a massive database, but a very slow bus speed, reproduction, and evolution. It knows almost everything about survival, but it takes thousands of years to retrieve a specific solution and implement it. The salt example. Despite millions of years of minerals washing into the sea, ocean salinity has remained at a steady 3.4% for nearly a billion years. Life, specifically microbial mats and geological sinks, pulls salt out of the water at the same rate it enters. The oxygen example. Atmospheric oxygen has stayed near 21% for 200 million years. If it rose to 25%, forests would spontaneously combust. If it dropped to 15%, large animals would suffocate. The collective mechanism, the superorganism. In a eusocial species, homeostasis happens at the ground level, often called social homeostasis. Honeybees maintain a constant hive temperature of 35 degrees Celsius or 95 degrees Fahrenheit, regardless of the weather outside. They do this collectively. In winter, they vibrate their wing muscles to create heat. In summer, they fetch water and fan it with their wings to create air conditioning. Termite mound structures are designed with lungs, tunnels that use wind pressure and thermal energy to maintain stable and humidity levels inside the nest. The mathematical fact. Negative feedback. Every homeostatic system requires three parts. A sensor detects the change, an example, a nerve or a chemical reaction. The control center, which processes the info, an example, the brain or the collective quorum. And then the effector acts to the fixed change, an example, a sweat gland or a phytoplankton bloom. Without homeostasis, a system experiences positive feedback where changes accelerate out of control, like a microphone screeching near a speaker, leading to system collapse, which is exactly what the Medea hypothesis argues happens in the long run. The following slide presentation explains the difference between neurons and bees and the entire Earth to explain how the globe is possibly conscious.
This video compares three levels of homeostatic systems: biological, eusocial, and planetary, using physical size and stability period. Planetary systems, like Gaia, are vast in size and stability, contrasting sharply with the small size and fast stability of biological systems. The firing rate equation relates a neuron's maximum firing frequency to its refractory period or reset time. For example, if the reset time is 1 millisecond, the maximum firing rate is 1000 Hz. Planetary homeostasis requires that the rate of change of a variable, such as CO2, be countered by a correction rate. This concept is modeled using a basic differential equation involving production and removal rates. Bremerman's limit defines the fundamental limit to how much information a physical system can process, based on energy and Planck's constant. Neurology systems have a high information rate, while Gaia's massive scale results in a slower information rate. The relativistic communication bound states that a system cannot be unified if its parts are further apart than light can travel during one firing cycle. The maximum diameter of the system is inversely proportional to its firing frequency. Applying these numbers, a human brain is well within the unity bound due to its small size and high frequency. Gaia's extremely slow cycle allows the entire planet to stay synchronized, despite its slow speed. This table summarizes the comparison. Neurology has a much faster clock speed and negligible relativistic lag, while Gaia operates on a geological timescale with a small but present relativistic lag. Homeostatic defense follows 3 stages to prevent collapse. Phase 1 is linear correction, where the system applies proportional counterforce. Phase 2 is nonlinear saturation, where buffers fill up and efficiency drops, leading to critical slowing down. Phase 3 is the tipping point, or bifurcation, where negative feedback turns into positive feedback, causing instability and collapse. The concept of a safety buffer can be visualized as a potential well. When the stress is too great, the system rolls into a state of collapse. Comparing natural and human-made homeostatic systems reveals key differences. Natural systems use DNA and ions, have high resilience, and are distributed. Artificial systems use code and steel, have low resilience, and are centralized.
While Lovelock did not originally claim Gaia was conscious, many researchers and philosophers have expanded the theory to include a collective consciousness. Some modern interpretations suggest just as neurons collectively form a human brain, the interaction of all living things, including humans and technology, could form a planetary intelligence. Recent studies in social neuroscience suggest that consciousness may have evolved specifically to solve social problems, such as predicting others' behaviors to stay together as a group. Julian Jaynes believed that consciousness did not evolve biologically, but was a cultural invention that developed from the breakdown of the bicameral mind. This is why I suggest that social problems have not been solved yet. Theories such as the noosphere suggest that the internet and digital networks are accelerating a shared human consciousness, effectively serving as a nervous system for the planet. The noosphere is the sphere of human thought, a third stage of Earth's development following the geosphere and the biosphere, characterized by the emergence of a collective human consciousness and mental activity that transforms the planet. Research often compares the collective behavior of humans to eusocial species, like ants and bees, which function as superorganisms. Evolutionary biologists like Richard Dawkins have argued that natural selection acts on individuals, not whole planets, making the idea of an evolved global purpose difficult to justify scientifically. While Richard Dawkins argues that natural selection acts only on the individual, the evolution of termites into superorganisms proves that biological individuality can scale upward when cooperation becomes the optimal survival strategy. If humans are inherently a social species whose collective potential has been suppressed by accumulated genetic trauma since the era of LUCA, then our current fragmented state is a biological malfunction rather than our true nature, because Earth is the only known planet to harbor life. It functions as a single self-regulating Darwinian individual. Therefore, the standardization prioritizing a global mental health is not merely a social goal, but the necessary biological intervention to heal the internal trauma of this planetary superorganism. Finally, allowing humanity to operate as a unified, functional component of the Gaian system. In direct opposition to Gaia, the Medea hypothesis suggests that life is inherently self-destructive and frequently triggers mass extinctions rather than maintaining balance. Humans are already developing technologies designed to mimic or support natural homeostasis at both the biological and planetary scales. While these systems aren't yet as efficient as 4 billion years of evolution, they use the same feedback loop principles discussed. To support the Gaia hypothesis of a self-regulating Earth, scientists are proposing Gaia 2.0, using technology as a deliberate regulatory layer. Companies like Planetary Technologies are developing antacids for the ocean to neutralize excess mirroring the natural carbon silicate cycle. Proposed technologies like stratospheric aerosol injection aim to reflect sunlight, acting as an artificial volcanic loop. though conspiracy theorists consider this to be nefarious. Near real-time monitoring through satellite networks and AI creates a global nervous system that tracks planetary boundaries like soil health and emissions. In medicine, we are moving from fixing problems to reverse engineering the body's natural balance. Technologies like continuous glucose monitors and automated insulin pumps function as a proportional integral control system, replacing the homeostatic role of the pancreas. Researchers are creating artificial cells that can collectively regulate their local pH, mimicking how living tissues protect themselves from environmental fluctuations. Scientists have built analog circuits modeled after thermosensitive neurons to create artificial temperature regulation systems for robots. While we can build individual loops like a thermostat or a dialysis machine, we have not yet replicated the extreme resilience of natural systems. Biological systems heal by redirecting internal energy to repair tissue. Most human tech is brittle. It cannot yet self-repair or spontaneously evolve new homeostatic loops when the environment shifts. As shown in our previous math, human supercomputers require massive power compared to the 20-watt homeostasis of the human brain. Research into the Gaia hypothesis and collective consciousness focuses on measurable feedback loops and emergent biological behaviors that allow complex systems to function as single units. The Gaia hypothesis posits that Earth is a self-regulating system where living organisms, the biota, influence the physical and chemical environment to maintain habitability. Life maintains Earth's atmosphere in a state of thermodynamic disequilibrium. For example, the persistence of reactive gases like oxygen and methane simultaneously is a result of continuous biological activity. Without life, these would react and disappear. The claw hypothesis mechanism suggests a negative feedback loop where warmer ocean temperatures increase the growth of phytoplankton. These organisms produce dimethyl sulfide, which enters the atmosphere and acts as seeds for cloud formation. Increased cloud cover reflects more sunlight, effectively cooling the planet. Plants and microorganisms regulate atmospheric homeostasis through photosynthesis, and by accelerating rock weathering, plant roots and soil fungi increase the surface area of rocks exposed to the air, which speeds up the chemical reaction that pulls from the atmosphere to form carbonate rocks. Daisy World mathematical simulation proved that planetary regulation does not require conscious purpose. In the model competition between black daisies, which absorb heat, and white daisies, which reflect it, naturally stabilizes a planet's temperature as its sun gets hotter, solely through individual growth rates and albedo changes. In scientific literature, collective consciousness is often studied as collective cognition or swarm intelligence, where a group performs tasks that no single individual can achieve. Eusocial superorganisms in colonies of ants, bees, and termites, individual members act as specialized cells. Data shows that these groups use distributed information processing to solve complex problems, such as finding the shortest path to food or selecting a new nest site through consensus voting. Recent research into insect brains has identified a central complex that supports spatial navigation and movement organization, similar to the vertebrate midbrain. Experts have declared a realistic possibility that many animals, including insects and octopuses, experience forms of subjective consciousness. The social complexity hypothesis suggests that consciousness evolved specifically as a tool for social coordination, In this view, the ability to model the self and others allowed individuals to survive better in large groups, eventually leading to the highly connected social structures seen in humans and other social species, when specifically focused on the evolution of consciousness as a tool to model selves and others. It is often referred to as the social interaction theory of consciousness, most notably championed by psychologist Nicholas Humphrey. The media hypothesis is a scientific framework proposed by paleontologist Peter Ward in 2009 that directly opposes the Gaia hypothesis. It argues that multicellular life is inherently self destructive and that is its own worst enemy. While Gaia posits that life regulates the environment to remain habitable, media suggests that biological activity frequently triggers mass extinctions and environmental collapse. Ward points to the geological record, arguing all but one of the Earth's major mass extinctions were caused by life itself, rather than external factors like asteroids. Research supporting the hypothesis cites several catastrophic events triggered by biological activity. The Great Oxidation Event. 2.45 billion years ago, early cyanobacteria evolved photosynthesis and released oxygen as a byproduct. This was toxic to the dominant anaerobic microbes of the time, causing a planetary scale poisoning and mass extinction. Photosynthesizing organisms repeatedly removed so much carbon dioxide and methane from the atmosphere that they triggered global deep freeze events, nearly sterilizing the planet. Ward argues that past warming events, often triggered by geological activity, caused oceans to stop mixing. This allowed anaerobic bacteria to thrive, which released lethal amounts of hydrogen sulfide gas, poisoning the atmosphere and destroying the ozone layer. The hypothesis predicts a terminal decline for complex life based on current biological trends: carbon dioxide depletion, photosynthetic organisms, and biologically driven rock weathering have steadily reduced atmospheric pressure over the last 200 million years. The 10 ppm limit. Ward calculates that roughly 500 to 900 million years, biological activity will drive levels below 10 parts per million ppm. At this concentration, plants can no longer survive, leading to the collapse of the entire aerobic food chain. Measurements of the geological record suggest that Earth's total biomass actually peaked roughly 1 billion years ago and has been on a long-term decline due to these Median feedback loops. Recovery is possible, though. It is factually true that microbes group together and exhibit collective behaviors that mimic a multicellular body. This grouping is not random, but is driven by sophisticated biological and chemical mechanisms. I suggest that the Earth utilizes Median feedback loops, where life-driven processes inadvertently create environmental crises, not as a means of self-destruction, but as a recursive filter that demands higher complexity for survival. These cycles of biological stress act as a transgenerational pressure, forcing life to evolve more sophisticated eusocial coordination to withstand the very instabilities it creates. In this view, accumulated genetic trauma is the biological record of these Median events. However, it is also the catalyst that pushes the biosphere toward a conscious, self-regulating state where prioritizing global mental health becomes the ultimate feedback loop to ensure long-term stability and complexity. I acknowledge Peter Ward's Medea hypothesis, but flip the script. The destruction is the engine of complexity. I am arguing that trauma isn't just a mistake, it's the environmental debt that humans must now pay by evolving into a more cooperative superorganism. It fits the unintentional requirement by suggesting Earth doesn't plan this. Rather, the mathematical necessity of surviving a Median crisis results in more complex systems. Most bacteria in nature do not live as a single free-floating cell, but instead aggregate into dense, highly organized communities called biofilms. Microbes secrete a sticky extracellular matrix of sugars, proteins, and DNA that glues them to the surfaces and each other. Within a biofilm, individual cells specialize in specific tasks, such as producing food, defending the colony, or focusing on reproduction, acting more like specialized tissues in a larger body. Living in a group allows microbes to withstand antibiotics and environmental stresses up to 1,000 times better than when they are alone. To coordinate these groups, microbes use a chemical communication system called quorum sensing. As I stated before, this is one of the things required for a global homeostatic balance, which is why I believe the Earth is a superorganism. Bacteria constantly releases small signaling molecules. When enough bacteria are present in one area, the concentration of these signals hits a quorum, a critical threshold. Once the threshold is reached, it triggers a simultaneous change in gene expression across the entire group. This allows thousands of individual cells to act at once, such as launching an attack on a host's immune system or beginning to glow like bioluminescence. Scientific research identifies several active collective behaviors in these groups. Wolf pack hunting, Myxococcus xanthus bacteria form predatory packs of thousands of cells that move together to overwhelm and digest other microbial prey. Some species use swarming motility, where cells differentiate into specialized swimmer forms and move across surfaces as a coordinated wave. Microbial groups can decide as a unit whether to migrate, defend, or even enter a state of dormancy based on pooled information from their environment. Some research describe these organized biofilms as a form of bacterial consciousness or intelligence, as the colony processes sensory inputs and produces complex adaptive outputs that no single cell would manage alone. The foundational building block for complex life was the emergence of sensory inputs and coordinating adaptive responses as a single unit. These bacterial colonies provided the first template for the superorganism, allowing life to survive early Median events that would have decimated isolated cells. This suggests that eusociality is not a late-stage evolutionary luxury, but a core survival mechanism encoded since the dawn of life. Consequently, modern human fragmentation is the regression from this ancient biofilm logic, caused by the compounding weight of genetic trauma accumulated across eons of environmental upheaval. While the researchers are still debating the exact timeline, recent genomic and geological evidence suggests that complex microbial ecosystems were likely already active 4.2 billion years ago. A 2024 study suggests that the last universal common ancestor, which lived roughly 4.2 billion years ago, was not a solitary organism. It likely lived in a recycling ecosystem where its waste served as a food for other microbes. This early ancestor already possessed a rudimentary immune system, a form of CRISPR-Cas to defend against viral invaders. This suggests that microbes and viruses were already interacting in a biological arms race. Shortly after Earth's formation, putative microfossils found in the Nuvuaqitu crustal belt in Canada contain tiny tubes and filaments made of hematite that resemble modern microbes that found at deep-sea hydrothermal vents. These structures are estimated to be between 3.77 and 4.28 billion years old. Evidence of biofilm formation, the sticky mats microbes used to group together, is robustly recorded in fossil record as early as 3.25 to 3.5 billion years ago in the form of stromatolites. However, many scientists believe biofilm formation was intrinsic to the very first life forms because surfaces like rocks and minerals provided the necessary stability for life to emerge. While single-celled organisms were the dominant form of life before the Great Oxidation event, the ecosystem was far more diverse than a single type of microbe. Life was divided into two vast domains, bacteria and archaea, each with distinct ways of surviving without oxygen. Research indicates that several specialized groups flourished in the ancient methane-rich oceans. These were likely the most common life forms. They breathed carbon dioxide and hydrogen and released methane as waste. They were the primary source of the Earth's early greenhouse gases before cyanobacteria evolved to release oxygen. Other bacteria use sunlight for energy but use chemicals like sulfur or iron instead of water, meaning they produce no oxygen. These microbes breathe sulfate, a salt found in seawater, and release hydrogen sulfide. They often competed with methanogens for resources in the seafloor. Surprisingly, recent research from MIT suggests that as soon as the first cyanobacteria began tiny oases of oxygen, roughly 2.9 billion years ago, other microbes evolved enzymes to gobble up that oxygen to use for energy. This happened hundreds of millions of years before oxygen actually filled the atmosphere. For most of the period before the GOE, life was strictly prokaryotic, simple cells without a nucleus. The complex cells that make up plants and animals did not emerge until the oxygen levels rose significantly. Current estimates place the origin of eukaryotes between 2.2 and 1.8 billion years ago, which is either during or just after the GOE. While fossil evidence for viruses is non existent, genetic analysis suggests that they have existed as long as cellular life, likely acting as a hidden force that drove the evolution of the first bacteria in archaea. In the hundreds of millions of years leading up to the Great Oxidation Event, oxygen was not yet in the atmosphere, but it existed in oxygen oases. These were localized pockets, primarily in the upper layers of the ocean, where oxygen levels were high enough to support early aerobic oxygen-using life. These oases formed directly around colonies with cyanobacteria because these microbes were the first to evolve oxygenic photosynthesis. They produced oxygen as a waste product. Oxygen would accumulate in thick microbial mats on the seafloor or in the sunlit photic zone of the open ocean. The rest of the ocean was filled with dissolved iron and methane, which acted as a sponge that immediately neutralized any oxygen that drifted too far from the colony. Geologists have found chemical fingerprints of oxygen in rocks that are 2.5 to 3.0 billion years old, long before the atmosphere became breathable. These metals only wash into the ocean when oxygen is present to weather them out of rocks on land. Their presence in ancient shales proves that oxygen was present in specific coastal areas. Scientists called the period roughly 50 to 100 million years before the GOE the whiff of oxygen, where these oases began to expand and leak small amounts of gas into the air. These oases were the world's first high-energy environments. Using oxygen for energy is roughly 15 times more efficient than anaerobic methods used by other microbes. This allowed life within the oases to grow faster and more densely. Genetic molecular clock data shows that the enzymes needed to process oxygen, like superoxide dismutase, evolved well before the GOE. This confirms that microbes were already adapted to live in these small oxygen-rich bubbles. From the perspective of the Medea hypothesis, these oases were the ground zero for the coming catastrophe. As these oases grew and merged, the oxygen eventually overwhelmed the planet's ability to absorb it, leading to the mass extinction of the anaerobic microbes that had ruled Earth for 1.5 billion years. The next major leap in forms was the eukaryote cells with a nucleus, which emerged roughly 1.8 to 2.1 billion years ago. From the perspective of connecting the dots to collective consciousness, this was a pivotal moment because eukaryotes are the literal result of microbes merging together forever. The transition from the simple bacteria to complex eukaryotes wasn't caused by a slow mutation, but by a process called endosymbiosis. A large host cell, an archaea, swallowed a smaller high-energy bacterium. Instead of digesting it, the two formed a permanent inseparable bond. That swallowed bacterium became the mitochondria, the power plant that exists inside every one of your cells today. This is the first biological fact of collective consciousness. Every complex life form on Earth is actually a team of once independent microbes that surrendered their individuality to become a single, more powerful unit. Billions of independent cells grouped together biofilms to act as one. Those groups become so tightly integrated that they evolved into a single new individual, multicellularity. Once these complex eukaryotic cells existed, they began grouping again, roughly 600 to 800 million years ago. These cells stopped just living near each other and began wiring themselves together via specialized proteins to form diverse animals like sponges. This is where the consciousness dot connects. In these new multicellular groups, cells needed to communicate instantly across long distances within the body to coordinate. These groups developed neurons. A mind or consciousness is effectively a collective agreement between billions of individual cells or neurons to process information as a single entity. 4.2 billion years ago, microbes grouped together for survival in biofilms like quorum sensing. 2.0 billion years ago, microbes merged internally to create complex cells, endosymbiosis, and 800 million years ago, microbes complex cells grouped together to form bodies, multicellularity. Multicellular individuals, humans, grouped together via technology and language. 4.2 billion years ago, there were no predators in the sense of animals with teeth or claws. Instead, predation happened at the molecular and viral level. Based on current genomic research into LUCA, the last common universal ancestor, and early Earth conditions, these were the primary predatory forces. Viruses are likely as old as life itself. Genetic analysis shows that even the earliest microbes had evolved CRISPR-like immune systems specifically to defend against viral predation. Viruses hunt by hijacking a microbe's machinery to replicate, usually destroying the host cell in the process. CRISPR clustered regular interspaced short palindromic repeats is a revolutionary gene editing technology that allows scientists to precisely edit, add, or delete DNA sequences within the living organisms. Adapted from a natural bacterial immune system that cuts viral DNA, it utilizes a guide RNA to direct a specialized enzyme, most commonly Cas9, to cut a specific DNA location, enabling gene inactivation or correction. While the fossil record for 4.2 billion years ago is sparse, later evidence suggests that some early microbes were epibiotic predators. These vampire cells would latch onto the outer membrane of other microbes, puncture the cell wall using specialized enzymes, siphon out the innards, nutrients, and energy of the host cell. In the early microbial world, eating was often synonymous with engulfment. One microbe would surround another with its cell membrane and absorb it. This wasn't always a kill, as discussed earlier. This specific form of predation eventually led to endosymbiosis, where the prey, like the ancestors of mitochondria, survived inside the predator. Life in the early collective colonies and biofilms was highly repetitive. Microbes produced antibiotic like toxins to poison neighboring species that were competing for the same chemical resources like hydrogen or sulfur. This is considered a form of interference competition, a precursor to active hunting. At 4.2 billion years ago, the environment itself was the greatest threat. High UV radiation, frequent meteor impacts, the late heavy bombardment, and extreme volcanic heat acted as selective pressures that hunted any organism not protected within a collective group or deep sea vent. By 800 million years ago, Earth was entering the Devonian period, a massive turning point where life shifted from slimy mats to complex visible organisms. This era provides the strongest facts on how individual cells began to form a collective biological unit. Before this point, almost everything was single-celled. Around 800 million years ago, we see the first definitive evidence of cells locking together permanently. The glue, cadherins. Genetic research shows that the proteins required for cells to stick together, cadherins, evolved or diversified around this time. This was the chemical handshake that allowed independent cells to form a single body, red and green algae. Complex multicellular seed weeds became widespread. These weren't just colonies, they had specialized structures for anchoring to rocks and for photosynthesis. Genetic molecular clocks and biomarkers, chemical fossils, suggest that the ancestors of sponges appeared around 800 million years ago. A sponge is essentially a collective of specialized cells. They have no heart or brain, but they coordinate to pump water and filter food. Fun fact, if you put a living sponge through a blender, the individual cells will find each other and reassemble into a sponge again. This is the ultimate example of a collective identity surviving the loss of the individual structure. With larger life came larger hunters. Single celled eukaryotes, protists, began evolving the ability to graze on bacterial mats. Vase shaped microfossils found in rocks dating to 750 to 800 million years ago. These are the remains of tiny shell building amoebas. They are the first evidence of the life building armor, which proves that something was finally big enough and aggressive enough to eat them. Around 800 million years ago, oxygen levels in the ocean began to pulse upward. Oxygen is highly efficient fuel. This extra energy allowed life to move beyond simple survival and invest in the expensive biological machinery needed for nervous systems and collective coordination. This era was the calm before the storm. Shortly after 800 million years ago, Earth entered this cryogenian period where the planet froze nearly solid. Some researchers argue that the rise of these new complex collectives, algae, and the early animals, pulled so much out of the air that they accidentally triggered this global deep freeze. After the Great Oxidation event, the next major global extinction occurred roughly 1.8 billion years later during the end Ediacaran extinction, approximately 542 million years ago. While the GOE itself is often called the oxygen catastrophe because it killed off the dominant anaerobic life forms, the period that followed, the Boring Billion, was categorized by relative biological stability. The next major extinctions was the pre-dinosaur and Ediacaran extinction 542 million years ago. This event wiped out the Ediacaran biota, the first large complex multicellular organisms, likely caused by an anoxic event, a drop in ocean oxygen, or biological recycling, where newly evolved animals, the first predators, ate or disrupted the older Ediacaran forms following the Ediacaran extinction. Earth entered the Phanerozoic Eon, which contains the famous Big Five mass extinctions. Late Ordovician, 444 million years ago, caused by intense glaciation and falling sea levels, killing 85% of marine species. Late Devonian, 372 million years ago, a prolonged series of extinctions often linked to algae blooms and ocean anoxia. Permian Triassic, 252 million years ago, known as the Great Dying, is the largest extinction in history, wiping out 96% of marine species and 70% of land vertebrates. Connecting to collective consciousness, the end of Ediacaran extinction is particularly interesting. It represents the moment when the stationary collective, life that looked like leaves or mats, was replaced by mobile collectives, animals with guts and nervous systems. This shift required a higher level of internal coordination and individual agency, setting the stage for complex brains that we see today. The Boring Billion, approximately 1.8 to zero point 0.8 billion years ago, was a period of extreme environmental stability that acted as a critical practice phase for collective reorganizations that laid the groundwork for collective consciousness by transitioning from the independent cells to integrated systems. One of the most significant grouping events in history occurred during this time, the origin of sexual reproduction. The red algae Bangiomorphia, dating to about 1.05 billion years ago, is the earliest known sexually reproducing life form. Sex is a fundamental shift from individual replication to a collective genetic strategy. It allowed superorganisms to mix their DNA and create a shared genetic pool that accelerated evolution far beyond what simple cloning could achieve. Research from the University of Sydney suggests that the breakup of the supercontinent Nuna, roughly 1.5 billion years ago, doubled the length of shallow continental shelves. These shallow seas became ecological incubators with stable temperatures and higher nutrient levels. In these crowded, nutrient rich zones, eukaryotic cells were forced into close proximity. This environment favored those that could coordinate their behavior, leading to more complex forms of multicellularity. The transition to collective existence, multicellularity, is expensive. A study from Arizona State University found that multicellular organisms require more than a tenfold increase in energy compared to single celled ones. Because oxygen levels were low, only 0.1% to 10% of modern levels, life spent this billion years slowly refining its internal metabolic processes. Scientists call this period the slingshot of complex life, where the slow buildup of biological innovations was quietly preparing for the explosion of complex animals later on. Life wasn't just in the ocean. The land was likely inhabited by cyanobacterial mats and early protolichens. By 1.2 billion years ago, these terrestrial microbial communities were abundant enough to affect global weathering and geochemical cycles. This presents a collective impact on the planet's physical state, a core tenet of the Gaia hypothesis. The first form of eusocial species, where individuals live in a colony with specialized castes, like workers and bees, appeared in termites approximately 150 to 170 million years ago, during the Jurassic period. While life had been grouping for billions of years, this was a distinct leap because individuals surrender their own reproductive rights for the benefit of the collective superorganism. Termites evolved from wood roach and cockroaches. Research shows that the shift to eusociality was driven by their diet. Wood is extremely difficult to digest. Termites rely on specialized gut microbes to ensure the next generation got these essential microbes. Offspring had to stay with their parents and eat the microbes from them, a process called proctodeal trophallaxis. This forced the family to together, eventually leading to a permanent multi-generational colony. The oldest definitive social termite fossils, such as Basisotermes, date back to the early Cretaceous, but genetic molecular clock data points back to the late Jurassic. Ants and bees appeared much later, around 120 to 140 million years ago, early Cretaceous. Ants evolved from wasp-like ancestors. Their success was tied to the rise of flowering plants, which provided new food sources and habitats. Most eusocial insects, ants, bees, and wasps, share a unique genetic system where sisters are more closely related to each other than they would be to their own children. The genetic fact made it biologically logical for them to help their mother, the queen, reproduce rather than doing it themselves. True eusociality is extremely rare outside of insects. The only known eusocial mammals are naked mole rats, which evolved this behavior roughly 20 to 30 million years ago. Like termites, they live in underground colonies with a single breeding queen and none reproductive workers. Eusociality is the ultimate biological expression of collective consciousness. No single termite knows how to build a 20-foot mound with a sophisticated ventilation system. The knowledge exists only in the collective interactions of the thousands of individuals. The individual is the colony. In these species, the individual insect is often compared to a single cell, while the entire colony is treated as the organism. Eusociality is fundamentally genetic, but it is triggered by a combination of specific DNA structures and environmental switches. In bees, ants, and wasps, a unique genetic system called haploidability makes eusociality more likely. The math. Because of how they inherit chromosomes, sisters share 75% of their DNA with each other, but would only share 50% with their own offspring. Evolutionarily, it is more efficient for a worker to help her mother produce more sisters, a 75% match, than to have her own children, which is a 50% match. This genetic quirk pre-wires the species for collective behavior. While the capacity for eusociality is in the DNA, the role cast of an individual is usually determined by epigenetics, changing how genes are expressed without changing the DNA sequence itself. In honeybees, all larvae are genetically identical. If a larva is fed royal jelly, it flips an epigenetic switch that activates queen-specific genes. The queen releases chemicals that physically suppress the reproductive genes in the workers. The workers have the motherhood genes, but they are chemically locked by the presence of the queen. In some ant species like fire ants, scientists have identified a specific genomic region, often called the social chromosome. A specific variant of the GP9 gene determines whether a colony will accept only one queen, monogene, or multiple queens, polygene. If you change this one genetic marker, the entire social structure of the collective changes. For a species to become eusocial, it must undergo a genetic trade-off. Over millions of years, eusocial species often lose genes related to individual survival, like certain immune responses or specialized foraging senses, because they rely on the collective's protection and food supply. If the instructions for the collective are written in the DNA, it suggests that collective behavior is a physical, heritable trait, not just a learned behavior. It means the group is a biological reality as real as a heartbeat. In mammals, specifically naked mole rats and Dammarlin mole rats, eusociality evolved through a perfect storm of extreme environmental pressure and unique biological constraints rather than the specific sister math haploidability found in bees. Mole rats live in harsh and arid regions of Africa where food, underground tubers, is extremely rare but massive when found. A single mole rat digging alone has a near zero chance of finding a tuber before it starves or is eaten by a snake. By grouping into a colony of 80 plus individuals, they can create a massive branching tunnel network. This collective foraging is the only way to find enough energy to survive the environment. Unlike most mammals that disperse to find mates, mole rats face high dispersal costs. Digging through bone-dry concrete like soil to find a new mate is often a death sentence. This led to high levels of inbreeding because the colony is so genetically similar. An individual worker is almost as closely related to the queen's offspring as it would be its own. This removes the genetic penalty for not reproducing. In naked mole rats, the queen maintains control not through royal jelly, but through physical dominance and stress. So the facts. The queen is the largest and most aggressive member. She subs the workers frequently. This physical stress causes a hormonal reaction in the workers that suppresses their reproductive systems. Their brains literally downregulate the production of sex hormones, GnRH, making them functionally sterile as long as the queen is present. This is a prime example of convergent evolution. Even though mole rats are mammals and termites are insects, they both arrived at the superorganism model because it was the only mathematically viable way to survive in their specific high-risk environment. In mammals, this shows that collective behavior can be forced by the environment until it becomes hardwired into the endocrine hormone system. The individual's brain is no longer independent. It is chemically enslaved to the status and presence of the queen. E.O. Wilson, the world's leading authority on social insects, argued in his later work, specifically The Social Conquest of Earth, that humans are one of the only eusocial mammals on the planet. He claimed our success is due to a biological glitch that forced us into the same superorganism path as ants and termites. To Wilson, the links between humans and collective consciousness is based on these biological facts. Wilson argued that eusociality always starts with a defensible nest. For humans, this was the controlled use of fire and permanent campsites. Once humans gathered around a fire to protect the group and cook food, the individual was no longer safe alone. Evolution began favoring genes that allowed us to read minds, cooperate, and protect the nest, rather than just ourselves. E.O. Wilson uses the phrase read minds to describe a uniquely human evolutionary trait known as intense social intelligence or mentalization, rather than telepathy. Wilson means the cognitive ability to create mental models of others' emotions, intentions, and thoughts in real time. This is Wilson's most famous and controversial factual argument. He stated that human evolution is driven by two opposing genetic forces. Individual selection favors selfish traits, competing for food and mates within the group. Group selection favors altruistic traits, cooperating so the group beats other groups. Wilson argued that our collective consciousness, morality, honor, and tribalism is a literal genetic result of group selection. Groups that grouped better survived, those that didn't die out. Wilson pointed to the wiring of the human brain. Unlike chimpanzees, humans have shared intentionally. Humans are the only primates that instinctively look where other person is pointing. This is the quorum sensing of the human species. Our brains are hardwired to sync up with others to achieve a collective goal, like hunting a mammoth or building a city. Wilson compared human language to the chemical signals and pheromones ants use to coordinate. Language allows us to create a virtual collective. We don't need to be touching to act as one unit. We can use words to align the consciousness of thousands of people across distances. Wilson's final conclusion was that a human city or a nation functions exactly like a colony. Just like termite casts, soldiers, and workers, human societies survive through extreme specialization. No single human can make a smartphone or grow all their own food. The knowledge is distributed across the collective. Wilson's core fact was this: humans are chimpanzees that have become eusocial. We have the selfish instincts of a primate, but the collective organization of an ant. E.O. Wilson's theory suggests that human eusociality played a significant role in our status as a single, global species by creating a united front that outcompeted and excluded other hominin groups. While several factors contribute to our biological unity, Wilson's framework emphasizes how our collective nature prevented the formation of subspecies. Wilson argues that the development of a nest, the campfire, and the subsequent extreme togetherness allowed Homo sapiens to dominate and preemptively exclude competing forms. Highly organized human groups likely acted as a united front, making archaic competitions like Neanderthals, who lacked equivalent large-scale social nests, as helpless as grasshoppers against army ants. By occupying a very broad ecological niche through collective labor and technology, humans outcompeted other similar species for the same resources, leading to their extinction. A primary factual reason we lack subspecies is that humans are too well connected and move around too much. Unlike other species that remain isolated in specific habitats, humans have a long history of migration and interbreeding across vast distances. Constant mixing through migration and trade acts as a biological blender, preventing local populations from being isolated long enough, typically requiring hundreds of thousands of years to evolve into distinct subspecies. Humans are a relatively young species with remarkably low genetic diversity, significantly less than our closest relatives, chimpanzees. Any two humans share approximately 99% of their DNA, which is not enough differentiation to justify biological subspecies. Around 900,000 years ago, human ancestors nearly went extinct, with the global breeding population dropping to as few as 1,280 individuals. This massive loss of genetic variety meant that the groups that eventually populated the world were starting from a nearly identical genetic foundation. In Wilson's framework, human subgroups are cultural ethnicities rather than biological subspecies. Wilson notes that while we are permanently conflicted between selfish and group impulses, we use culture and language to negotiate these differences, maintaining the species' integrity through social cooperation rather than biological divergence. Eusociality is not entirely absent from the ocean, but it is extremely rare. While terrestrial environments support thousands of eusocial species, like ants, bees, and termites, only one marine genus is known to exhibit this behavior. Snapping shrimp of the genus Synalipheus model, far more difficult to achieve in the ocean than on land. Eusociality requires a shared nest where generations overlap and care for young together. Most marine superorganisms use broadcast spawning, releasing eggs and sperm into the water column to be carried away by currents. This massive dispersal makes it nearly impossible for family groups to stay together long enough to form a colony. The eusocial snapping shrimp are among the few marine species with direct development, meaning their larvae hatch as crawling juveniles that stay in the same home sponge rather than floating away. A defensible nest is a factual prerequisite for eusociality. Water is roughly 800 times denser than air, exerting massive lateral pressure. Complex and self-built nests, like paper wasp nests or large mounds, are more likely to buckle or be destroyed by shifting sediments and currents. Marine environments lack the lignin found in wood that terrestrial insects use to build durable, multi-generational tunnels. Without wood or soil that can be easily glued together, marine life must rely on the existing cavities like sponges, which are in short supply and fiercely contested. Maintaining a large crowded colony creates significant metabolic demands. Oxygen diffuses much slower in water than in air. Pumping fresh oxygenated water through a large complex nest is orders of magnitude more expensive in energy than in air-based ventilation. Crowded colonies in confined spaces risk suffocation or hypoxia, which limits the activity levels needed to maintain and defend a nest. In terrestrial systems, extreme temperatures and unpredicted food sources force individuals to group together for survival, like the arid food problem in mole rats. The ocean's high heat capacity means temperature fluctuations are less extreme, removing one of the major pressures that typically drives the evolution of protecting nesting. Open water acts as rich feeding ground for young organisms. This provides a strong evolutionary incentive for offspring to leave home and seek food elsewhere, rather than staying to help the collective. Schools of fish are explained by collective behavior, or shoaling, but they are biologically distinct from eusociality. While a school of fish acts like a single unit, it is not a superorganism because every fish remains a fully independent, reproductive individual. There are three factual pillars that explain why fish school. Proposed by W. D. Hamilton, this theory explains that fish don't group together to help the collective. They do it to reduce their own individual risk. If you are in a group of 1,000 or your individual chance of being targeted by a predator is 0.1%, predators that hunt by sight, like marlin or tuna, find it difficult to track a single target when hundreds of similar looking bodies are crisscrossing their field of vision. The predator's brain experiences sensory overload. Just like cyclists in a peloton, fish in a school use energy saving fluid dynamics. Fish utilize the vortices, swirls of water, created by the fish in front of them. This allows the trailing fish to hitch a ride on the moving water, reducing the energy needed to swim by up to 20 to 30%. Fish constantly shift positions within the school so that no single fish remains at the high drag front for too long. The instant synchronized turns seen in schools are not directed by a leader or a group mind. Instead, they are governed by local rules and a specialized organ called the lateral line. The lateral line is a series of pores along the fish's body that detects tiny changes in water pressure. Computer models, like Void's, show that schooling emerges from just three simple rules followed by each individual. Don't bump into your neighbor, steer in the same average direction as your neighbors, and stay close to your neighbors. In the context of collective consciousness, schools of fish represent distributed intelligence, a system where complex global patterns emerge from simple individual interactions without any central command. The reproduction only definition is a major point of scientific tension. While the classic Crespi and Janiga definition, 1995 insists on permanent, irreversible sterility for a species to be truly eusocial. Many modern biologists argue this is too rigid. The debate centers on whether eusociality is a binary switch, you are or you aren't, or a spectrum of collective behavior. Many researchers argue that defining eusociality based on the sterile caste ignores the degree of sociality in other species. They use terms like subsocial or semisocial to bridge the gap. Some bees can choose to be a worker or go off and start their own nest depending on environmental cues. If they can switch back, the classic definition says they aren't eusocial, but their behavioral coordination is identical to those that are. If we only count sterile species, we exclude thousands of species that behave as integrated collectives, but keep their reproductive options open. There is a massive debate between kin collection, the math of sharing genes, and group selection, E.O. Wilson's focus. The reproductive focus, supporters of kin selection like Richard Dawkins, argue reproduction is the point. The collective only exists because it's clever way for selfish genes to copy themselves. Supporters of group selection argue that coordination and labor division are the more important facts. They argue that once a group acts as a superorganism to survive, the reproductive status of individuals is just a secondary detail. This is where the sterility rule breaks down most famously. Humans are clearly the most socially complex species, yet we have no sterile caste. E.O. Wilson argued that humans represent a new kind of eusociality driven by culture. We have functional castes, people who choose not to have children, monks, celibate leaders, or career-focused individuals, but contribute to the colony's survival. Some biologists call this cultural eusociality. In some species, like orcas and humans, females live long past their reproductive years, menopause. While they aren't born sterile, these individuals become a dedicated non-reproductive class that manages the collective memory of the group, finding food during droughts, raising kin. If these grandmothers are essential for the group's survival, they function exactly like a sterile worker caste in an ant colony. To mathematically explain how collective behavior sociality develops without mandatory sterility, researchers use models that weigh the cost of individuality against the benefits of the group. In short, an altruistic trait will favor kin selection if the benefit to the relative, adjusted for their relatedness, outweighs the cost of the individual. This is the foundational equation for the kin collection. If the benefit to your relatives is high enough, your genes win even if you never reproduce yourself. This allows for helper roles in a collective without requiring a sterile caste. Multi-level selection theory, championed by E.O. Wilson, uses a nested equation to explain how collectives form: individual selection, competition within the group. This favors selfish individuals who reproduce more. Group selection, competition between groups. This favors groups with more cooperators. When the survival pressure between groups is stronger than the internal competition, the group begins to act as a single superorganism. In this model, everyone can remain fertile, but they choose not to compete internally because the group survival is the only way for any individual to
Hamilton's
survive.
rule describes the conditions for an altruistic gene to spread through a population, RB greater than C. This rule considers the coefficient of relatedness, R, the sum of benefits to the recipient, B, and the fitness cost to the actor, C. The multi-level price equation is the primary mathematical framework for multi-level selection. It partitions the total change in a trait into within-group and between-group components. These components include between-group selection, within-group selection, and the average fitness of the entire population. Key mathematical models include MLS1 versus MLS2. MLS1 defines group fitness as the average fitness of individuals in the group. MLS2 defines group fitness by the proliferation of the groups themselves, such as how many new offspring groups are formed. The fixation condition for cooperation suggests that cooperation is favored by multi-level selection if the benefit-to-cost ratio satisfies a specific inequality involving the maximum group size and the total number of groups. Multi-level selection and kin selection are often mathematically equivalent. MLS explains the evolution of altruism when the variance between groups exceeds the variance within groups, which aligns with the relatedness requirement in Hamilton's rule. The price equation is the mathematical foundation for describing how any trait changes in a population over time. One of its two components is the selection component, which measures the covariance between a trait and fitness. The second component is the transmission component. This represents the average change in the trait value between parents and offspring. It accounts for factors like mutation, environmental shifts, or biased inheritance. Finally, here are the definitions of the variables used in the price equation. $delta bar $z is the change in the average trait value of the population. $W underscore I is the fitness of individual $I. $bar W is the average fitness of the population. $Z underscore I is the trait value of individual $I. And $delta Z underscore I is the difference between offspring and parent trait values.
The price equation is the mathematical holy grail for this research. It breaks down how a trait like collective consciousness changes over time, how much a trait helps an individual succeed, how much the trait changes within the individual over time. It proves that if group beneficial traits provide a large enough advantage during group-on-group conflict, those traits will spread through the entire species, even if they are slightly disadvantageous to the individual. Evolutionary game theory. Mathematical simulations of the prisoner's dilemma show that in population of selfish actors, the only way to reach a stable state is through iterated cooperation. I help you, you help me. Indirect reciprocity. I help you, someone else sees it and helps me later. This is the basis of reputation. In large groups like humans, these mathematical interactions create a social web that functions as a collective consciousness, regulating behavior through shared rules and peer pressure rather than biological sterility. Engineers and roboticists use the exact same mathematical frameworks to design satellite swarms and autonomous robots as biologists use to study ants and bees. While the physics of satellites involves orbital mechanics, the logic of their collective behavior is built on the math of social evolution. Hamilton's rule in robotics in biology. This rule predicts when an individual should sacrifice its own reproduction to help a relative. In robotics, engineers use it to program altruistic decision-making. Instead of genetic fitness, robots use task productivity as their fitness metric. A satellite might sacrifice its own battery life to act as a data relay for another satellite, provided the total health of the swarm improves. Price equation for evolving algorithms. The price equation is used in swarm robotics to understand how collective traits change over time. This is used to model how an algorithm or a strategy is passed from one robot to another during a mission. Engineers use this to partition a swarm's performance into individual learning versus collective adaptation, helping the swarm evolve its behavior in real time to survive harsh environments like deep space. Distributed intelligence versus central control. Just like a school of fish or an ant colony, satellite swarms are designed to have no leader. Each satellite follows simple local rules. An example, stay kilometers from neighborhood A. The collective consciousness of the swarm emerges from these simple interactions, allowing thousands of satellites to coordinate perfectly with the single human or master computer giving every command. Market pricing for resources to manage limited resources like fuel or bandwidth. Satellite swarms often use game theory and price mechanisms inspired by social insect foraging. Satellites bid for tasks based on their current fuel levels, creating a self-regulating market that maximizes the swarm's overall lifespan. If we apply E.O. Wilson's framework to modern humanity, the biggest obstacle to a fully integrated eusocial-like structure is the tension between individual selection and group selection. Wilson famously described this as the iron rule of social evolution. Selfish individuals beat altruistic individuals, but groups of altruists beat groups of selfish individuals. Here are the specific factual obstacles preventing us from functioning as a true superorganism. The cheater problem in individual selection. In a true eusocial colony like ants, cheating is rare because individuals share nearly identical DNA. Helping the group is helping yourself. Humans are genetically diverse and driven by individual fitness. In a large-scale collective, free riders, individuals who take resources without contributing, have a massive advantage. To prevent the collective from collapsing, humans have to expand enormous energy on policing, laws, and surveillance, costs that an ant colony doesn't have. Scalability in the Dunbar limit. Our brains evolved for small group collective consciousness, not global systems. Dunbar's number suggests humans can only maintain stable social relationships with about 150 people. Once a group exceeds this size, we can no longer read minds or trust everyone instinctively. We lose the biological glue of the collective and have to replace it with abstract systems like money, flags, religions, which are prone to corruption and misunderstanding. Subgroup competition and tribalism. The same group selection that makes us cooperative also makes us aggressive toward outsiders. Humans are wired for parochial altruism. We are kind to our nest, but hostile to others. Instead of forming one global Gaia-like superorganism, humans tend to form competing superorganisms, nations, corporations, and ideologies. This leads to resource wars rather than a single coordinated effort to manage the planet. The loss of shared reality for a collective to act as one, it needs a shared sensory input, like the pheromones in a beehive. Digital technology has created algorithmic silos. Instead of collective consciousness, we have fragmented echo chambers. When different parts of the human superorganism cannot agree on basic facts like climate, safety, and economics, the system experiences a form of autoimmune failure where the parts attack each other. Biological autonomy, unlike the snapping shrimp or the mole rat, humans still have reproductive and physical autonomy. Evolution has not yet locked us into specialized castes, because every human can theoretically survive and reproduce independently. We constantly face the urge to defect from the collective to prioritize our own family line. We have the technology of a superorganism, but the biology of a hunter-gatherer. We are caught in an evolutionary middle ground where the costs of being an individual are high, but the costs of fully surrendering to the collective are also terrifying. If humans fully integrated into a social superorganism, the possibility of new discoveries would likely shift from individual breakthroughs to systemic leaps that are currently impossible due to our fragmented nature. Massive scale parallel processing in a eusocial structure. Research would function like a distributed computing network. Currently, scientific progress is slowed by the silos of individual careers and the need for researchers to spend years catching up on existing data. A fully integrated species could process data across millions of minds simultaneously. This will likely lead to an intelligence explosion in fields like protein folding, climate modeling, and fusion energy, where the complexity of the variables exceeds the capacity of any single human brain or current AI. Radical resource reallocation, one of the biggest scientific hurdles today, is the cost and competition for resources. Roughly 2% of global GDP is spent on R&D, often duplicated by competing nations or companies. A superorganism would eliminate the waste of competition. We could mobilize the collective labor and wealth of the planet toward a single massive goal, such as terraforming Mars or building a Dyson swarm around the sun, with the same singular focus that a termite colony uses to build its mound. Biological and genetic mastery. If the species moved toward a eusocial caste system, we would likely discover new ways to engineer ourselves. Eusociality often results in specialized bodies, an example, soldier ants versus worker ants. Integration would remove the ethical and social barriers to germline genetic engineering. We might discover new forms of human biology, such as individuals specifically designed for deep space travel, or hyperfast computation, leading to a diversified biological toolkit for the exploring of the universe. Direct neural communication, the noosphere. Integration would require a biological or technical glue, like a brain-to-brain interface. Language is a low bandwidth communication tool. We lose roughly 90% of the concept when we try to explain it in words. Direct neural integration would allow for the collective experience of abstract concepts. We might see higher dimensional mathematics or feel quantum states, leading to a new era of quantum physics and metaphysics that our current individual sensory systems cannot perceive. However, there is a factual downside. History shows that biodiversity and cognitive diversity are the engines of resilience. If the human superorganism becomes a monoculture of thought, we might lose the lucky accidents and contrarian views that lead to a paradigm shift like Einstein's relativity. Like a colony that succumbs to a single virus, a fully integrated species might lose the ability to discover solutions to novel, unforeseen threats. From a biological and evolutionary standpoint, the cost of surrendering to a collective is measured by the loss of the individual fitness and systemic vulnerability. If humans were to fully integrate into a social-like superorganism, the specific costs would include reproductive forfeiture in every known eusocial species. The primary cost is the surrender of the right to reproduce. Evolutionarily, your success is measured by passing on your specific genes. In a collective, most individuals become genetic dead ends, working solely to ensure the queen's or the central system offspring survive. This would mean the end of the nuclear family and individual choice in lineage, replaced by a centralized, optimized breeding program. I argue that human superorganisms as a neural-style network rather than an insect-style colony. In this model, eusociality doesn't mean becoming identical. It means becoming hyper-specialized and deeply connected. A superorganism is a monoculture that lacks diversity. The human brain is a superorganism of 86 billion neurons. It is the most resilient and creative structure known precisely because of its units that are specialized in visual, motor, and emotional, and it's not identical. True eusociality in humans would create a monoculture. It would create a cognitive ecosystem. Just as a biofilm's strength comes from specialized niches, global mental health allows individuals to express their unique contrarian brilliance without the static of survival trauma. We aren't becoming a single cell, we are becoming a global neural network where diversity is the primary source of processing power. The graph illustrates the steady decline in birth rates as millennials, born in 1981 through 96, and Gen Z, born on 97 through 2012, moved into their prime childbearing years. According to data from the Pew Research Center and the CDC, several key factors contribute to this downward trend. Many millennials and older Gen Z individuals are delaying marriage and parenthood to focus on higher education and career stability. Rising costs of housing, childcare, and student loan debt have made the financial entry price of parenthood feel unattainable for many. Recent surveys indicate an increasing number of young adults that are choosing to remain child-free by choice, prioritizing personal autonomy or lifestyle flexibility. As the oldest members of Gen Z enter their mid-20s, early data suggests that they are following the millennial trend of lower birth rates compared to previous generations at the same age. Prioritizing global mental health ensures that every individual's cognitive DNA contributes to the survival of the planet, making the genetic dead end argument obsolete in any age of digital and cultural immortality. Summary synthesis. The transition to a human superorganism does not require the loss of individual, but the transcendence of the ego. By healing accumulated trauma, we move from a state of defensive individualism, which actually creates more monoculture through tribalism, to a state of differentiated cooperation. Like a biofilm or a brain, our resilience comes from our connection, not our conformity. This perspective frames the declining birth rates seen in the graph not as demographic crisis, but as a deliberate evolutionary pivot. Under this summary synthesis framework, the data relates to the decline in the following ways. The decline represents a shift in how millennial and Gen Z generations spend their reproductive energy. Instead of biological output quantity, they are investing in the collective information, pool technologies, art, and social systems that's designed to sustain the biosphere. From K selection to Gaian selection, traditional biology uses K selection, fewer offspring, high investment. This theory suggests we are moving toward Gaian selection, where success isn't measured by how many children you have, but by how your cognitive DNA, ideas, and contributions ensures the survival of the planet. Healing defensive individualism. The declining birth rate may be a symptom of moving away from the survival at all costs tribal mindset. By having fewer children, individuals may be attempting to break cycles of accumulated trauma, choosing differentiated cooperation over the pressure to simply propagate the species. Redefining the genetic dead end. In this view, a millennial or Gen Z individual without children is not a dead end. If they contribute to the global mental health or the biofilm of human knowledge, they are achieved a form of cultural immortality that is more vital to the superorganism than biological reproduction. In short, the graph doesn't show a generation failing to reproduce. It shows a species transcending the ego to prioritize the resilience of the whole over the expansion of the parts. So as I repeat for emphasis, prioritizing global mental health ensures that every individual's cognitive DNA contributes to the survival of the planet, making the genetic dead end argument obsolete in an age of digital and cultural immortality. Summary synthesis. The transition to a human superorganism does not require the loss of the individual, but the transcendence of the ego. By healing accumulated trauma, we move from a state of defensive individualism, which actually creates more monoculture through tribalism, to a state of differentiated cooperation. Like a biofilm or a brain, our resilience comes from our connection, not our conformity. More downsides include loss of adaptive flexibility. Individual diversity is a biological insurance policy against change. Collectives thrive on standardization. For a superorganism to be efficient, its units must be predictable. If the environment changes suddenly, a new virus or a climate shift or a solar flare, a standardized collective is far more likely to suffer a total system collapse. A group of diverse, selfish individuals is more resilient because some will likely have the rare traits needed to survive the new threat. Cognitive homogenization, echo chambers. In a collective, the signal must be clear to coordinate millions of units. This requires the suppression of noise, which in humans is dissent, original thought. You lose the lone genius of the contrarian who challenges the status quo. Innovation in a collective is excellent at incremental optimization, making a better mound, but terrible at paradigm shifts, deciding to leave the mound entirely. Again, I counter this with the biodiversity of the mind, neural diversity. In biological systems, the most advanced superorganism, the human brain, does not function through standardization. It functions through extreme differentiation. A healthy human superorganism doesn't want identical units. It requires specialized cells. The artist, the engineer, the contrarian. If everyone thinks the same, the brain of the superorganism stops working. Therefore, cognitive diversity isn't noise to be suppressed. It is the very processing power that makes the collective intelligent. Anti-fragility versus efficiency. The downside, too, assumes the collective seeks efficiency, which is brittle. You can argue that a conscious superorganism seeks anti-fragility, a term coined by Nassim Taleb. Unlike a beehive, a human collective based on differentiated cooperation uses the connectivity to share locally discovered solutions instantly. If a lone genius finds a way to survive a new virus or climate shift, that rare trait isn't limited to their biological offspring. It is uploaded to the collective information pool immediately, making the whole system more resilient than a group of disconnected individuals ever could be. The edge of chaos principle, complexity science, shows that the most resilient system exists at the edge of chaos, the sweet spot between total order, stagnation, and total randomness, collapse. Dissent and original thought are the evolutionary mutations of the collective mind. A truly advanced superorganism would protect its contrarians because it recognizes that paradigm shifts are necessary for long-term survival. In this model, the lone genius is the superorganism's way of scouting the future. Digital immortality as a safety net, the loss of adaptive flexibility, argument relies on biological DNA, which takes generations to change. By shifting to cognitive DNA, we can adapt at the speed of light. We don't need to wait for a selfish individual to be born with a lucky mutation. We can intentionally innovate our way out of our threats. The collective doesn't eliminate the mound lever, it provides them with better tools to find the next home. Vulnerability to centralized parasites. A superorganism is only as good as its brain or its signal system. If the central command, the queen, the government, or the AI is corrupted or hacked, the entire collective follows the error to its own destruction. As discussed with the Medea hypothesis, life often creates its own worst enemies. A human collective could be tricked into suicidal behaviors, like resource exhaustion, much faster than a group of independent skeptical individuals. Ethical and subjective erasure from a philosophical perspective, backed by social science. The cost is the loss of the self. Human happiness is tied to autonomy and agency. In a true social structure, your worth is defined strictly by your utility to the group. If you become injured, old, or inefficient, a superorganism's logic dictates you should be discarded or recycled for the good of the whole. I argue that the distributed intelligence model, the parasite argument, assumes a top-down hierarchy, a queen or a central AI. A truly evolved superorganism functions like the internet or a mycelial network, not a kingdom. There is no single point of failure. In a decentralized system, if one node is hacked or corrupted, the rest of the network recognizes the error as autoimmune stress and isolates it. Skepticism and dissent aren't bugs in the system. They are the white blood cells. By prioritizing global mental health, the collective ensures that its units remain discerning, preventing the suicidal groupthink seen in lower-ordered social structures. Countering ethical erasure, intrinsic versus utility worth, the recycling the elderly argument applies to mechanical systems, not conscious ones. In a superorganism based on cognitive DNA, an individual's worth is not tied to physical labor, but to their unique perspective. An injured or elderly individual carries a lifetime of data and accumulated wisdom that collective needs to navigate long-term cycles. In this framework, selfhood isn't erased, it is upgraded. Your agency is found in your unique contribution to the collective consciousness. We don't discard the old parts of the brain because they don't process as fast. We value them for the complex patterns they recognize. The holonic defensive borrowing from the philosopher Ken Wilber, you can argue that humans are holons, entities that are both a whole in themselves and a part of a larger whole. Evolution doesn't work by destroying the previous level. It transcends and includes it. Cells didn't lose their individual chemical complexity when they formed organs. They gained a more stable environment to exist in. The self doesn't disappear. It is liberated from the defensive individualism of survival mode, allowing for higher order happiness based on connection rather than competition. From utility to resilience, a system that discards its inefficient members is actually less resilient because it loses the fringe data those members hold. A conscious superorganism recognizes that compassion is a survival strategy, not a luxury. By caring for the weak, the collective preserves a wider range of genetic and cognitive possibilities for future crises. Psychological research into de-individuation, the state where a person loses their sense of self in a group, reveals that being absorbed by a collective significantly alters human behavior and brain function. The breakdown of self-regulation when humans lose their identity in a group, their internal ability to self-monitor effectively shuts down. The brain shifts its focus from internal values to external group cues. This makes individuals highly responsive to the immediate vibe or emotional energy of the crowd. Anonymity in a group creates a diffusion of responsibility. Individuals feel less personally responsible for the group's actions because they believe they cannot be singled out or blamed. As the self-awareness fades, a person's personal moral standards become less influential. This often leads to disinhibited actions, behaviors the person would normally find unacceptable or even illegal, such as aggression or vandalism during riots. De-individuation is a primitive, low-level state. The superorganism is a high-level conscious state. Traditional de-individuation occurs when people are anonymous and unconscious. In a conscious superorganism, the goal isn't to hide the self in a crowd, but to make the self fully transparent. With digital trails and cognitive DNA, anonymity disappears. When an individual feels seen and connected to the whole, their sense of responsibility doesn't diffuse, it expands. You aren't responsible for just your actions, you feel responsible for the health of the biofilm. From external cues to internalized global values, the argument assumes the vibe of the crowd is impulsive or aggressive. If the superorganism is built on global mental health and healing trauma, the external cues being broadcast aren't calls to riot. They are signals of cooperation, empathy, and long-term sustainability. The brain isn't shutting down self-regulation, it is outsourcing the heavy lifting of moral computation to a more sophisticated collective ethical framework that is more consistent than a single stressed individual's brain. Psychologists like Mihaly Csikszentmihalyi describe flow as a state where the self-consciousness fades, but performance and creativity peak. Being absorbed by a collective doesn't have to be a downward move into mob violence. It can be an upward move into a collective flow state. Like musicians in an orchestra or players on a high-performing team, the loss of ego awareness allows for a level of complex synchronized art that no individual could achieve alone. This isn't disinhibition toward aggression, it's optimization toward creation. Integration as the cure for defensive individualism, the disinhibited actions of riots often stem from unresolved trauma and a feeling of powerlessness. The superorganism model explicitly prioritizes healing trauma. When the individual feels safe and integrated, the defensive need for aggression vanishes. De-individuation in a riot is a cry for help from a fractured system. Integration in a superorganism is the resolution of that fracture. Behavioral contagion and the group mind, early theorists like Gustave Le Bon argued that crowds create a mental unity or crowd mind. In a de-individuation state, people become more suggestible, taking on the motives and behaviors of those around them almost like a puppet. Emotions like fear, anger, or excitement spread rapidly through the group, amplifying collective behavior at the expense of personal restraint. De-individuation is a primary driver of behavior in online spaces. The lack of face-to-face feedback and the use of anonymous usernames reduces empathy. This explains why people who are otherwise law-abiding might engage in cyberbullying or trolling. It's not always negative, though. There is pro-social de-individuation. While often linked to mob mentality, de-individuation can lead to positive collective action if the group's norms are positive. Collective resilience. During crises such as the 7/7 London bombings, de-individuation fostered a shared identity of "we are in this together," leading to spontaneous, coordinated altruism among strangers. Community bonds. Group activities like communal dancing, sports celebrations, or peaceful protests can build a powerful sense of solidarity and shared purpose. To conclude this video, most of this is data, and I will be showing the references. Some may say that my hypothesis is not credible, but I am just having a lot of fun doing the research. What I do want to say is that the downsides of what the legal structure would be is extremely scary. But I believe that a eusocial society can still exist without the loss of freedoms. What makes us different as humans is our ability to find value in anything and anyone. Just imagine affordable childcare, the ending of world hunger, and as I was researching this, possibly even the war. It is easy to assume that this is only achievable through laws and a way of life that is unacceptable, but that is only because this is the type of power structure that we have been living in since the start of governments themselves. That is why I find mental health to be the very answer to all the suffering that we see today. If accumulated genetic trauma suppresses human eusociality, then prioritizing global mental health serves as a global biological antidote. By utilizing evidence-based therapies like problem-solving therapy at scale, we can modulate epigenetic markers of stress, effectively re-unlocking our species' latent capacity for selfless, large-scale cooperation. The solution to the human failure to operate eusocially is the standardization of global mental health practice. By treating mental well-being as a national development priority, we reduce the survival-based hypervigilance that leads to tribalism, allowing for the division of labor and social cohesion characteristic of advanced social species. Studies suggest that epigenetic markers like DNA methylation are not permanent. Positive conditioning and therapeutic interventions can reverse the biological scarring of trauma. Prioritizing the mental health of youth and parents yields intergenerational benefits, potentially breaking the cycle of inherited trauma that has accumulated since ancient times. Experts note that social connection is a critical factor in mental health recovery. Thus, a eusocial structure, high community support, and mental health are mutually reinforcing.
The Gaia hypothesis, proposed by James Lovelock and Lynn Margulis, suggests Earth is a self-regulating system that maintains conditions for life. The foundational text is Gaia, a new look at life on Earth from 1979, with The Ages of Gaia, 1988, offering a more detailed scientific biography. Countering this is the Medea hypothesis, coined by Peter Ward, which argues that multicellular life is inherently self-destructive and often leads to its own extinction. The primary source for this counter theory is Ward's 2009 book, The Medea Hypothesis: Is Life on Earth Ultimately Self-Destructive? Sources on mass extinction include Peter Ward's Under a Green Sky, which explores how past global warming triggered greenhouse extinctions. A. Hallam's Catastrophes and Lesser Calamities examines the varied causes of Earth's Big Five die-offs. For Microbiology Foundations, Microbiology: An Introduction by Tortora, Funke, and Case is a standard comprehensive textbook. Essential Microbiology by Stuart Hogg offers a clear entry into the diversity and applied roles of microbes. Major works by E.O. Wilson include Sociobiology, The New Synthesis, which explores the biological basis of social behavior. Consilience argues for uniting sciences and humanities. And The Superorganism examines insect colonies as integrated units. References on collective consciousness include the terms Origin by Emile Durkheim and Peter Godfrey Smith's Other Minds on the biological evolution of awareness. Gödel, Escher, Bach discusses consciousness distributed across multiple organisms, like ant colonies. Bonnie Bassler studies how bacteria use chemical signaling to coordinate collective behaviors. J. William Costerton, the father of biofilm science, introduced the term biofilm. Eshel Ben Jacob pioneered the study of microbial intelligence, suggesting bacterial colonies exhibit, quote, social intelligence. Armin Jalali connects stigmergy and chaos theory to describe biofilm as bacterial consciousness. Antony van Leeuwenhoek made the first historical observation of biofilms. Rachel Yehuda is a pioneer in epigenetic trauma research, studying Holocaust survivors. Elizabeth Binder researches the FKBP5 gene, a stress response switch altered by early life trauma. Isabel Mansoui shows trauma symptoms can pass down generations via small RNA. Michael Meany and Moshe Jaif proved that maternal care quality can permanently change gene expression related to stress response. E.O. Wilson coined eusociality. Other researchers modeled how cooperative structures evolve and how trauma can be addressed. Catherine Panterbrick directs a program focused on how socio-emotional learning and resilience building can mitigate the biological impacts of trauma in refugee populations.
In the next video, I will look at human prehistory and how the trauma of prehistoric life keeps biting us in the brain. Thank you and see you next time.